This presentation will go over some of the terminology and importance of cell culture methods. Cell culture is the process of maintaining living cells in a laboratory environment, which is referred to as in vitro. This process allows for controlled experiments to occur and has several advantages. One of the most important considerations is that cell culture greatly reduces the need for animal studies, which would be referred to as in vivo. Laboratory animals require specific conditions, and many do not consider it ethical to use animals for research when there are other viable options. Of course, cell culture cannot replace the need for animal studies entirely because a whole organism is much more complex than a single type of living cell. At some point, many therapeutic treatments must be tested in animals before testing in humans can commence. But studies in cell lines often come before the animal studies. Because cell lines are one cell type, they provide a well-controlled, homogeneous environment to study the effect of various molecules and cell components without the interference with the complex whole animal studies. Of course, cell culture has disadvantages as well. Cells growing in culture are not exposed to the myriad of chemical, hormones, and other molecules circulating within an organism. It's really impossible to completely recreate an in vivo environment in vitro, so cells may not react in the same manner they would if they were in vivo. This reinforces the idea that whole animal studies will need to be employed at some point during the research of a specific effect. Remember that the genotype, which is the genetic makeup of the cell, often differs in cell lines grown continuously in culture compared to normal versions of that cell in an organism. And genotype affects the phenotype, the appearance and behavior of cells. The phenotype is what most often is being investigated in cell culture research. When you add or do something to the cell, what happens? That something is usually a phenotypic change. Let's discuss the different types of cell lines for cell culture. A primary culture is the culture of cells taken directly from an organism and grown in the laboratory in a flask, plate, or dish. A secondary culture is when you take the primary culture and pass them to fresh environment, a new flask, dish, or plate. Usually these types of cells will only replicate a certain number of times before they die out. They have a finite number of replications and then they act old and die no matter how you try to nurture them in the lab. The process of cell dying in this manner is apoptosis, a programmed, controlled cell suicide. This is in contrast to a process of cell death called necrosis, where the cell sort of just breaks open and dumps everything out. This is not a controlled and usually results in damage to nearby cells. To make a primary culture, you must remove sterile tissues aseptically. The tissue is cut into small pieces and then subjected to an enzymatic digestion to separate the cells from one another and from the non-cellular matrix to which they were attached in the organisms. The cells are then added to a culture vessel with media with the appropriate nutrients. Hopefully they grow, at least for a time, so that you can do your experiments. A cell line is a type of culture that has undergone mutations and will not undergo apoptosis after a specific number of divisions. These cells will divide forever, provided they are given the appropriate nutrition and space to do so. They are immortal. Often cell lines are composed of transformed cells. These are cells that have been altered from the normal state by a virus or chemical mutagen. These changes make the cell line immortal and will often cause tumors if injected into a specific animal host. A hybrid cell line, or hybridoma, is a cell line created after a specific cell type of interest was fused to a transformed immortal cell line. Not all cells survive this unusual process, but those that do are maintained if they display a phenotype of interest, such as the ability to produce a specific protein of interest. Cell lines are described as adherent or suspension. Adherent cells grow while stuck to the bottom and sides of the plate or flask. Suspension cell lines grow while floating in the medium. To pass adherent cell lines, they must be treated with trypsin so that they will release from the side of the vessel. The main cell line we use during this course is an adherent cell line derived from mouse fibroblasts. Fibroblasts are cells that are common in connective tissue and secrete collagen and other proteins to help create connective tissue. It's categorized as a biosafety level 1, so it's not a dangerous cell line to work with. We will continue our discussion on cell lines in part 2.